Hello, everyone. Welcome to the 2020 Forest Health Meeting, our revised format where we are online instead of in person, and hoping that this works for a lot of you. It looks like we are currently up to 64 attendees, so some of you were able to get this to work. We have a very packed agenda for today, so we're going to try and do several updates on the most relevant forest health topics that we have in Ohio this year in brief 15-20 minute periods. So we're going to try and move through these fairly quickly and try to get it to work for everybody here. But before we get started, everybody should have access to a Q&A box. So it should be a little speech bubble with a question mark. We're going to use this to communicate for the meeting today. So this is the only option we have to talk back and forth between our presenters and our participants. If you need continuing education credits for either Society of American Foresters or International Society of Arborists, what I need you to do is in the Q&A box, please type in a question that includes your full name, whether you need ISA or SAF credits, and your member ID number. This will document that you are here for this meeting. I will probably ask you to do the same thing at the end so that we have a timestamp of when you were on and when you logged off. So what I need you all to do is in the Q&A box, please type in your full name, whether you need ISA or SAF credits, and your member ID. And we'll see if we get those to come in. And I believe that we have a little bit of a delay between us actually presenting and you receiving it. So when we do question and answers and things like that, we will probably pause for a little bit to make sure that we are getting everything in before we move on to the next speaker. But we are going to start out today with our first speaker, who is Tom Macy with Division of Forestry, and he's going to give an update on white pine needle issues. So Tom, if you want to go ahead and share your screen, I will try to switch over our feed. And I'm going to ask that everybody has some patience with me as we work through this new online format, try and do this meeting. Hoping it works well, but we're going to see how it goes. So let's see if we can get Tom live. And you should be good, Tom.
Great. So you're able to see my video and see my presentation now? Okay, great. Okay, so again, thanks everybody for joining and for bearing with us with any technical difficulties. Hopefully folks are able to get on okay and can view the updates. And I wish we could have seen everybody in person at the Forest Health meeting this year, but we're doing what we can to get these updates out to you. So I really appreciate Stephanie's help in getting this all set up. We're all learning as we go along here. So I'm going to give an update on eastern white pine health in Ohio. And so I'll get into that. We've got some issues with white pine in different parts of the state here recently. So we've had a major increase in reports of eastern white pine discoloration. And those really started to ramp up in about 2018. I noticed some discoloration. We got a few reports coming in of this issue prior to that, but 2018 seemed to be really the turning point with a lot of these white pine problems that we're starting to see. Most of the reports are coming in from the southeastern part of the state. There are reports from elsewhere, but that seems to be where the majority of this is occurring. And these images here sort of show the symptoms that we're seeing on white pine. So in most cases, what we're seeing this on forest-grown plantation of white pine, planted white pine. Although there are reports of this also from yard and landscape trees and open-grown white pines. like the one you can see there on the right. So, in general, we can see this this discoloration happening, this sort of graying or browning of the foliage, and not that typical dark green or bluish green coloration that you see in a healthy white pine. So I wanted to show this map. Hopefully you can see that. There are some small red areas on the map that we can see there. And these are polygons that we drew and mapped from our annual forest health aerial survey. So the red polygons that you're seeing on the map are areas that were either labeled from the air or checked later on the ground and labeled as white pine needle damage. And white pine needle damage is the main issue responsible here. I'll talk more about that. So those are shown here on the map, but what's also shown in these red polygons are polygons that might have been labeled as unknown conifer discoloration, defoliation, or mortality. So it's possible some of these aren't white pine, but there are other conifer issues. The 2019 data was pretty much all ground checked and verified, but the 2020 areas haven't fully been checked on the ground yet. But these are the areas that we've mapped in the last 2 years. So that totals over 12,000 acres, and most of that was this year. And that's 438 total polygons mapped. And again, most of that was mapped this year. And the 2020 ground checks are not yet complete. So that the map just kind of shows you where we're seeing most of this, at least from our aerial survey, which we fly in June. And so you can see the concentration of these is mostly in the southeastern part of the state, with also some going up into the northeastern part. So what have we done to look into this problem? Back in November of 2018 and June of 2019, we did do some field investigations into this. We had a group of plant pathologists and forest health experts, a couple pathologists from U.S. Forest Service, state and private, and also the West Virginia Department of Agriculture. Their plant pathologist came up and joined us. So we had staff from Division of Forestry, Ohio Department of Agriculture, and Wayne National Forest staff all meet up and kind of look at some of these sites to see what's going on with the white pine. And so we did, in 2018, we visited Zaleski State Forest, and in 2019, it was at Wayne National Forest, the Athens district. So we examined the trees in the field and collected samples for lab analysis. We also, in 2018 at Zaleski, we put the drone up. We have a UAV drone, and that's what the image on the right is, an area at Zaleski State Forest that's seeing a lot of that white pine needle damage. And so there's a view from the air of what some of those pines are looking like. A lot of them are looking pretty bad. So the main findings of these investigations, the impacted trees had lost all but the current year's needles, and the remaining year's needles are discolored. So white pine needles typically last 1.5 years, but they were dropping early with only this year's needles remaining. So you can see that there on the image on the left, and the image there on the right are some pretty heavily impacted white pines at Zaleski. Lower branches tend to be more heavily impacted, and we'll talk a bit more about the diseases impacting them, but in a lot of cases, the spores of the fungi are moved in rain splash, and so the very upper canopy usually isn't very heavily impacted, and the lower branches tend to be more heavily impacted. So a variety of native fungi were identified on the foliage, both in field investigations, looking at the needles, as well as lab investigation. And this is a list of some of those fungi that were identified on the needles. So they seem to all be playing a role here. Some of them may be more prevalent than others in most cases, but it seems to be a variety of these fungal diseases infecting the needles that's killing them and causing this early defoliation. And so this photo on the right from Danny Martin, pathologist with the Forest Service, is one of the needles with the fungal fruiting body starting to erupt from the needle there, taken under a microscope. So we have this suite of fungal pathogens that is collectively known as white pine needle damage. This white pine needle damage seems to be on the rise in the last few years across the whole range of eastern white pine. There is this fact sheet that University of Massachusetts has put out recently. I believe this is back from 2016, but it describes the issue that they're seeing in New England, which is really similar to what we're seeing here in Ohio. So there's other issues with eastern white pine that do not appear to be major factors here in Ohio, but some of these are pretty serious problems of white pine in other parts of its range. But here in Ohio, it seems to mainly just be this white pine needle damage. I wanted to mention a few of these publications that provide a lot more information on white pine needle damage. This 2015 publication identified the main pathogens that are involved. The 2018 publication discusses the amount of defoliation caused by white pine needle damage, and it showed that most of this defoliation is happening in the summertime, and these white pines then are experiencing decreases in growth because they're not doing as much photosynthesizing because they have less foliage on them throughout the growing season. So they found a 25 to 73% reduction in basal area increase on those trees. And then another 2018 paper is a synthesis of emerging health issues of eastern white pine. So possible reasons for this, quote unquote, explosion of white pine needle damage that we're seeing in different parts of the state. Really, I think the main thing is weather conditions. So we've had a lot of moisture in recent years, and this figure here is the Palmer Modified Drought Index going back to 1950, and it basically shows where we have wetter than normal years or drier than normal. Wetter than normal represented by those green bars, and you can see just here in the last few years there's a lot more green bars than yellow. And 2019 in particular was extremely wet year, and so I think all this additional rainfall and humidity is allowing these foliar fungal pathogens to really proliferate. Host plant stress. So when you have a stressed tree, it becomes susceptible to more pest and disease issues. And so I think in some cases, these white pines are planted on a site that isn't very conducive to really great white pine growth. You've got sites that may have poor soil drainage or high pH, sites where eastern white pine typically doesn't thrive. And so I have a note there also, young trees can be more resilient to these factors. So, it's possible that, white pine planted in a not great site could survive pretty well when they're young, the first couple decades of their growth, maybe even longer than that. And now they're getting to an age where the site just isn't great for their growth. Weather's been really wet, and you've got a stressed tree and conditions conducive to this fungal growth. And then competition-induced stress, and that can be seen in these unthinned plantations. So a lot of times we have, these white pine plantations that were planted back in the 40s, 50s, 60s, and they haven't been managed, and the trees are really crowded. They've got small crowns. They're not getting enough sunlight or space to grow. And so that can induce stress on those trees as well. So you've got a stressed host plant, you've got these wetter conditions that are great for fungal growth, and so you get this this white pine needle damage really doing some damage. And just want to mention the native range of eastern white pine. So we are on the fringes. You know, a lot of southern Ohio is not truly in the native range of eastern white pine. So they may be in sites that aren't great for their growth. And just wrapping up here. So, you know, what's the outlook and what are potential management options? Really, it comes down to quality white pine management. There is this manual that recently came out, field manual for managing eastern white pine. This is for New England, but I do think a lot of the principles would apply here in Ohio. So I'd be happy to point folks to that. Stand thinning of eastern white pine plantations can improve air circulation and can reduce white pine needle damage. There are some studies to show that it can reduce the damage caused by these fungal pathogens. In some cases, you may have trees that are too far gone. And so typically what you're looking for are leaving those trees that have a greater than 25% live crown ratio, so they can improve their health. If they have smaller than that, they likely won't recover. Obviously, removing hazard trees in high use areas is important. So the outlook is uncertain. Climate is going to be a major factor. You know, if we continue to have these wetter than normal growing seasons, it's unlikely that this white pine needle damage will get better. And so, you know, there's some predictions going out into the future with a changing climate that predict, you know, periods of pretty high rainfall during the growing season and also punctuated with some periods of drought. So it's tough to say, you know, what's going to happen, what the fate of these trees will be, but I think a lot is going to depend on the weather. So that's it for me. I'm going to wrap up, and I think we have a couple minutes left for some questions, if we have that, and then we'll get moving on to the next speaker.
Thanks, Tom. If anyone has any questions, please put them in the Q&A box. We'll give it a minute just to make sure we get those. I did have a question come in asking if this session was being recorded for anyone who couldn't attend. This will be recorded. Once we finish today, we'll figure out how to make it available to anyone who wants to look at it. So yes, this is being recorded, and yes, it will be made available later on. While we wait to see if we have any questions, next up is going to be Dave Adkins. So Dave, if you want to go ahead and share your screen, I can swap out the presenters. Tom, you may need to stop sharing yours.
Yeah. Did it come up? No, I don't see it.
Not yet. I do not see any questions coming in. And I still don't see your presentation there, Dave.
Yeah, I'm trying to get it up here.
Again, sorry guys, we would much rather have this in person, but get to deal with technology today instead.
Did you see the request from Ann Bonner asking for the list of fungal pathogens? I don't know the best way to get that to her.
Nope.
But.
Yeah, definitely. I can share that with. I'll try to put it in the Q&A area, but if not, I can definitely just shoot me an email and I can share that with you.
Well, where'd it go?
Let's see, we do have a few other questions. So I have a question, has there been damage reported on young plantations?
Yeah, I would say the majority are mature, overstocked, mature white pine. I don't know if we have the data to say what the ages of the pine that are affected typically are. I would say in most cases it does seem like they're larger, mature stands that are affected by this. But I have seen some relatively younger stands, but by and large it's the larger, more mature stands.
Okay, and then are there notable insect issues that are associated with the white pine needle diseases?
I'm not aware of any, associated insect infestations with white pine needle damage. It does just seem to be those foliage problems. But certainly, with a stressed host plant, you could invite additional attack by other pests, but I haven't seen a whole lot of that.
Are the pathogens native or non-native?
They're native pathogens. So they've been here. It's not like they're any new introduced diseases. They're native, and I think it's just a matter of a conducive environment, all this rainfall, and potentially in some cases a stressed host plant.
All right, and then we have an anonymous question if the tree farm committee will be providing dinner tonight. The tree farm committee is providing whatever you want to cook yourself tonight. That is one of the major drawbacks to having this online, I know. All right, well, thanks, Tom. And it looks like we are good to go here, so we're going to go ahead and switch to Dave Atkins with the Ohio Department of Agriculture to give an update on gypsy moth. And you are good to go, Dave.
Thank you, Stephanie. It's a pleasure to come before everyone and give a little update of what we're doing and what's been going on with gypsy moth here in Ohio. So we'll get started here. This is basically our 2020 treatment areas and project boundaries that were determined by the decision algorithm that we utilize at Virginia Tech. Also, we utilize Michigan State University for some data collection and also technical support with our program. But the DA at Virginia Tech is where everything is sent to be analyzed, both our treatments and trap catches, to determine where our potential problem areas are. Breakdown for 2020, we placed out 12,649 traps in 79 of Ohio's 88 counties. Our catch this year was a little less than last year. We had 29,000 last year. This year we had 26,000 moths. So it was down a little bit from the previous year. Total treatments that were conducted in Ohio amounted to 70,612 acres. 6% of that was larvicide, and 94% was mating disruption. The product we're using now for mating disruption is Splat GMO, which stands for Gypsy Moth Organic. It is a material that has the consistency of a hand cream. It's not the plastic flake anymore. So it is a new product that's in its third or fourth year of use. First couple of years we were testing it only on partial treatments, but now we've gone to full use of it. So it is something that has moved us forward. It'll be interesting to see how well this mating disruption worked this year. It's important that the product disperses the pheromone over what we shoot for is an 8-week period during the mating season to disrupt that mating process. But this year we had in excess of 21 days of 90-degree weather. So that quickens the dispersal of the dispar lure. And we're hoping that it didn't fizzle out on us before the mating season was over. So we're looking forward to next year's moth catch to see if we had any effects from the excessive heat we had here in the state this year. So we'll see what happens. Keeping our fingers crossed. 18 of the 79 counties that were trapped showed increases in moth catches over 2019 versus 2020. 11 of those are in our STS action area. So we'll be taking a lot of action on those areas for possible treatments. The rest of them are in what we call the generally infested area, and they fall under our suppression project, which is a voluntary project where the landowners must submit an application to have their properties treated and so forth. So, but we'll be working here in the next few weeks. We just finished up pulling traps. In fact, I was out yesterday pulling traps, and we'll be analyzing, sending all that data to Virginia Tech to be analyzed, and we'll be working, Brian and I will be working on getting our plans ready for 2021. So that's all in the upcoming days. To take a little look at history, we're looking at moth catches from the time the STS project started and when our treatment started in 2000. And we averaged over that 21-year period about 30,000 moth catches a year. And you'll see that it was pretty much a roller coaster ride, about 2-year spikes in the moth catches up until about 2012. And then everything sort of went under the average. And the one peak there in 2018 may be tied to the fact that we did not do our mating disruption on a timely manner. When we were treating, we like to get that treatment out just prior to the emergence of the adults. And in 2018, they were already out and flying when we started our mating disruption. So we had a little problem with the feds getting here on time and getting that done. So, but I think the trend is definitely in our favor. It's going down, and we're keeping it down. So we're looking good. Treatment acres, again, we average about 80,000 acres a year. The last 3 years, it's been just a little bit under that. We seem to have peaks about every 2 to 3 years. This last one was about 6 years, 5 to 6 years. So we hope we're stretching those peaks out a little bit more. And I think it also has to do with that same chart that Tom just showed about moisture levels and stuff. So we'll see what happens here, but it's looking good. I think we're keeping it under the. We're slowly going down below that 20-year average of 80,000 acres. So in the last couple years, it's been a little bit less than the 80,000. I think it's 79,000 last year and 76,000 this year. So we're looking good. Overall spread rate here in the state of Ohio, again, it varied up and down prior to 2010, and then after that, we've kept it under that zero advancement or equal to. So again, until 2018, when I think that our late treatment may have affected our potential to hold that down the spread rate. So the spread rate over the entire state has averaged out to a minus 4.3 kilometers per year over the past 20 years. So that's a little over 2.5 miles a year going backwards. We're sending it back to Pennsylvania is what we're working on. So hopefully, I keep telling my counterparts over in Pennsylvania they're going to be forced to come back into the STS program because I'm pushing it back to them. So In Ohio, we've got two zones that the decision algorithm works with on spread rates, and this is the zone 7, which is Northwest Ohio. Zone 8 is in Southeast Ohio. Northwest Ohio is mainly farmland and scattered forest areas, and Southeast is mainly contiguous forest areas. So there's a little difference in the spread rates between the two areas. Treatments are a little harder up in zone 7, Northwest Ohio, because the forests are scattered. There's a lot more open riparian areas. There are narrow riparian areas and fence lines, which gypsy moth can advance forward through. So, and they're a little bit harder to treat. So it has been moving, but we're still getting good results. In zone 7, we're averaging around minus 2.76 kilometers per year over the last 21 years. So we're still getting a good push back. Slow but sure. In zone 8, which is southeastern Ohio, where we have the more contiguous forested areas, we've been a little more successful. We've been in these areas, we can isolate the core populations better and hammer on a little bit better than we can in the open farmlands of Northwest Ohio. So in zone 8, the average spread rate has been a minus 5.87 kilometers per year over this time period. So still very successful. The maps here show our action zone from 2020 to 2019, the map on the left there. We were able to push it back just a little bit, about 6.818 kilometers this past year. So we had a little bit better success than we thought we were going to, but it's still success. The map on the right shows where the action zone was in red. They're back in 1999 when we started the program, and then where it is here in 2020. We've been able to move that zone back around 86 kilometers, or about 53 plus miles from where we began the STS program. So we feel very good about our program. It has been successful and continues to be successful in controlling the gypsy moth and pushing it back. And I think over the past several years, it hasn't been all just our treatments, but also the wet springs and so forth, where the Entomophaga myaga fungus has developed, and it's also helped to knock down these growing populations. But overall, I'm very happy with our program, and I think we have been successful and are continuing to be successful. So that's about all I have with our update for the gypsy moth. If there's any questions, here's my contact information about our program. If anyone who lives or works in the suppression area, and you got property owners that want to get together and put together a suppression block, please have them get in touch with us. Any questions?
Thanks, Dave. We do have one question so far.
Okay.
What are other states doing to suppress gypsy moths? When I lived in Michigan, their DNR had a stated policy of considering gypsy moth a naturalized pest and did not invest resources in control and reduction. Is this the approach other states are pursuing?
We are part of an 11-state integrated pest management program that runs from North Carolina all the way up to Minnesota along the leading edge. Michigan is totally infested with the gypsy moth, and they've pretty much raised the white flag on it. The other 11 states that are along the leading edge are very active in doing the same process that we've been doing here in Ohio, as far as treating these small populations that are developing in the transition zone and trying to hold steady with the advancement. Other states ahead of this line are very interested in what we're doing. I know Missouri is involved with us. Tennessee is involved with us. Kentucky. These states are all very concerned about the gypsy moth moving into their states. So they're very supportive of our program and of the efforts that the National Gypsy Moth program is performing.
Okay, our next question is, what size do you need to have a suppression block?
Okay, the criteria for a suppression block is a minimum of 50 acres contiguous forested area, at least 50% canopy, 35% preferred species in there. It does have to pass a T&E assessment with Fish and Wildlife to make sure there's no threatened or endangered species in there. And then it is a 50-50 cost share between the property owners and the state.
Okay, so a 50-acre block for multiple contiguous landowners? That was another question. And then what is the suppression zone? Just a clarifying question.
Let me go back here. It's everything north of the blue line there on the map on the right. Those counties are in what we call the suppression area. The area in between the two blue lines, that is our action zone. These are where populations are just starting. To the north and east of that blue line is where populations have established. So, and then below that blue line area is what we call the eradication area. Any populations that usually pop up there are due to human movement of one of the life stages. Whereas the transition is the natural transition of the moth westward. I can't hear you, Stephanie.
Sorry, I had it muted. Okay, so then we have what are the per acre costs to spray? They realize they might change, but the latest 2020 charge.
Usually for 2 apps of BT, it'll cost the landowner about $35 to $40 an acre. And then if you wanted to use the Mimic, it's about $30 or so an acre for the landowners.
All right. Well, that is all the questions I see for now. So why don't you go ahead and stop sharing your screen?
Okay.
And we're going to go ahead and let Todd share his screen next. We're going to rearrange the schedule just a little bit because we're having some technical difficulties with Phil. So Todd, are you able to go ahead and share your screen?
Just okay, are you seeing it now?
Not yet.
Yeah, I think that.
All
hopefully
right,
that
now
did
we
it.
should be good. It is transitioning now, and you are up.
Okay, thank you, Stephanie. And you can hear me okay?
Yes, I can hear you fine.
Okay. Well, good afternoon, everyone. Todd Hutchinson from the U.S. Forest Service. and I'm going to be giving a brief update today on Ailanthus wilt study that probably a lot of you are familiar with, that Joanne Rebick started about 6 years ago. And so I would just like to acknowledge my co-authors or collaborators on this. Really, Lauren Pyle is in Missouri. She's really taking the lead on this part of the project, and Leela Pinchot is also working with us. And then we're still consulting with Joanne as we move forward and try to continue at least parts of the research that she started before she retired. Okay, just some acknowledgments here before I get started. Again, just acknowledging Joanne. Without her, this project would have never gotten started. And funding for it was provided by the USDA Forest Service Forest Health Technology Enterprise team out of Morgantown, West Virginia. A lot of collaboration early on from Penn State and West Virginia, Don Davis and Matt Casson and others who were working on the Ailanthus wilt. Really like to thank the land management partners who allowed us to establish plots on their land, Division of Forestry, also the Wayne National Forest and the Wilds. And then really there's been a tremendous amount of field and lab work also by many people, but it's been led by Bill Borovica, Tim Fox, Danny Gill, Joan Jolliffe, who did a lot of the lab and culturing of the fungus, and Dave Runkle. All right, so just a little timeline here about the Ailanthus wilt and mortality caused by this Verticillium non-alfalfa. It was first really observed that there were large patches of Ailanthus wilting and dying in Pennsylvania and central Pennsylvania back in 2002. And by 2009, Don Davis at Penn State and some of his students had investigated and found that the organism that was linked to that wilt was, they identified it as Verticillium alboatrum, a fungus, a fungal pathogen that causes vascular wilt. And a few years later, it was the taxonomic name was changed to identify it as Verticillium non-alfalfa, which is not really that closely related, but looked a lot like the alboatrum. In 2009, then, after some extensive searching, Ailanthus wilting and the organism were found in Virginia. And then in 2012, Joanne had really put out the feelers in Ohio for foresters to look for this. And Matt Malone found a population in Pike County of wilting Ailanthus that did turn out to be the Verticillium non-alfalfa in 2012. And then Joanne applied for the funding to set up inoculation plots in the field using the Ohio fungus, the Ohio strain. And that's really what I'm going to be focusing on today, is those plots that were established back in 2014. Okay, this really, there's a lot of potential for this to be used as a biocontrol for Ailanthus, which is just such a widespread weed tree that causes a lot of problems in forests. It's a native soil-borne fungus. It's really cosmopolitan, but it's found, I think it's found in a lot of places in the eastern U.S. It just hasn't been found on Ailanthus. It's highly selective for Ailanthus. It basically kills Ailanthus by clogging the vascular system, preventing the uptake of water and nutrients. Comes in through the roots if it's not inoculated by scientists. It spreads also by natural root grafts from trees that form individual trees of Ailanthus that are in stands, and then also it spreads to clonal sprouts of the same individual, because as you know, Ailanthus can form a lot of clonal sprouts. It's persistent in the soil for at least 4 years. Some think it may even persist in the soil for upwards of 10 or more years. And it really, there's been a lot of testing of effects on non-target species. And there are some species that are affected when they're inoculated, but there's very few species that have been shown to be infected in stands of dying Ailanthus, where the fungus, rather than being injected, has to actually make it through the root barrier and to infect the tree that way. One example is devil's walking stick. Aurelia spinosa does have some susceptibility to the wilt, but for the most part, all the significant tree species have been tested and are not susceptible to this fungus. And it can be applied with tools that are already pretty readily available. The fungus can be cultured fairly easily in a lab. Concentrations of the spores then can be injected, just like you would an herbicide with the hack and squirt method here. That this picture is showing a tree being injected with the fungal inoculant there at the base of a ailanthus tree. And it really works quick. After a tree is injected, this is 8 weeks after an inoculation, it's already wilting, leaves are starting to defoliate, and then it's totally defoliated. And for the most part, most of the trees are dead after this point. There's could be an added bonus as far as the spread. It generally spreads fairly slowly in patches of ailanthus because it's going from tree to tree in the root system. But there may be a means where this ambrosia beetle, which is also non-native, and it does feed on dead and dying ailanthus. It might be transporting it from stand to stand of ailanthus. It's still not really well worked out, but it could be helping to spread it. So the study that I'd like to bring you up to speed on was done at 5 study sites in Southeast Ohio at Tar Hollow, Perry, and Blue Rock State Forests, also the wilds and some plots on the Marietta unit of the Wayne National Forest. At each one of these sites, Joanne established 5 plots, which were about a 1/4 acre, 50 meters by 20 meters. 4 of those plots were ailanthus stems, were inoculated, and in one they were not, just served as a control. And those inoculations were done in the summer of 2015, and about 10 ailanthus trees per plot were inoculated. I'll talk a little bit more about how many ailanthus trees were on the plots. So 10 were inoculated, and this is the range. There was quite a range in density of ailanthus on the plots. These are just the plots that were inoculated. So 4 at each site, just 3 at the wilds where we had to drop one of the plots. But you can see that there were generally about 40 ailanthus trees greater than 2.5 inches per plot. That's about 160 per acre. There was quite a range though, from as low as 13 up to about 134 at one plot at Tar Hollow. So they did range in density. And ailanthus, generally the basal area of these plots, the mean basal area was about 111, and ailanthus made up about 34% of the basal area. So they weren't totally monocultures of ailanthus, but ailanthus was a dominant stem in most of those, a dominant tree in most of those plots. And the trees, they generally, these were not huge trees that were on the plots or that were injected. These are the size of the injected stems. The average was about 5.5 inches, so they were pole-sized trees. Most of them were 2.5 to 8 inches or so in size that were inoculated back in 2015. And this, you can see that immediately those trees that were inoculated, the 10 per plot. This is 108 days, so the first fall after the June injections. There was a range. There was, especially at Tar Hollow and Perry, more than 90% of those trees were already dead or greater than 90% defoliated by the first fall. At Blue Rock, it was sort of intermediate at 78%, and then at Marietta in the wilds, there was a little bit less mortality or heavy defoliation. But the average was about 3/4 of the trees were either dead or heavily defoliated just in that first year. And this is what we see over time now. We've continued to track the trees on the plots. The graph on the left there is the injected stems. You can see that there only about 13% of those are alive today. So there were 188 trees that were that were injected. But we do have, there's a few trees, primarily at the wilds, that are still healthy, and we're not exactly sure, just the inoculations just didn't take as well. And then you can see on the right here, the non-injected stems, those trees that were just on the plots but were not injected. It is definitely spreading pretty quickly to those trees. Only 34% of the non-injected trees are still alive. And most of those, I should say on both of these, that 13% that is alive of the inoculated and the 34%, a lot of those have had wilt or are having wilt. They're not totally dead yet. And as we move on, looking at the differences in the sites again, the wilds, for some reason, those injected stems, we still have 40% survival. Most of the other sites are pretty similar, from 5 to 15% survival. And for the non-injected stems, similarly, the wilds, it hasn't spread as much on those plots. It has spread, but not as much. 60% of those non-injected stems are still alive, compared to 20 to 30% on the other 4 sites. And when we look by size, this makes sense that as for the injected trees, basically there's a greater proportion of the larger trees are still alive. The smaller trees are almost all dead that were injected. Just there's more fungus that's being injected into the tree, into a smaller tree. And that also goes for the non-injected stems. Almost half of the trees that were injected, that were 11 inches or bigger, that were injected or were not injected, are still alive today. And that percentage is much lower for those smaller 3, 5, 7 inch sized trees. Okay, other observations. I was on some of the plots this summer helping to collect data, but have not been on all of the plots. But I'll just give some other observations. I'm not really going to present any more data. There's definitely spread, though, that we've documented outside of those quarter-acre plots. So I wouldn't say it's moving lightning quick, but it is moving outside of those plots. We've documented over 200 trees on those treated plots that are outside of that quarter acre that have shown signs of wilt or are dead, and some of those are pretty large trees. I think for the most part, these plots were not set up in huge patches like 5 acres of ailanthus, though. So the plots, there may not be a whole lot of ailanthus stems outside of that quarter acre plot. They were sort of centered on small to medium patches where ailanthus was pretty common. So that may be limiting the spread eventually, is that it's just not a continuous stand of ailanthus that goes on forever. There's very little ailanthus regeneration or ingrowth, resprouting or sap. A lot of the either seedlings that have established or the sprouts that have come up after the tree has died have also gotten the wilt and are very, very weak and have either died or have signs of serious wilt. So the ailanthus seems to be having a low regeneration potential. There are some trees, though, where we've documented where they were at some point during the 5 years, they were totally defoliated, and then they have refoliated with healthy leaves. Some have refoliated, and those leaves have caught in the wilt, and I'm not sure if it's just a matter of time or whether there might be a little bit of resistance on some of these trees. Time will have to tell, but that's a very small percentage. And as far as what's coming back, as the death of all these ailanthus has opened up some pretty big canopy gaps. These stands, I didn't really mention it, but there's not a lot of desirable tree regeneration as far as oak hickory in these stands. I would say that there's very, very little oak was in these stands to start with. There was yellow poplar, sugar maple. There were more sort of mixed hardwood type sites. And really what we see responding, probably most common, at least on the plots that I've been on, have been the shrub layer. Spice bush is thriving. We have, pawpaw, multiflora rose in some places. It kind of depends on, whatever vegetation was there to start with, but we're not seeing a lot of regeneration of desirable tree species at this point. There has been a lot of ash regeneration on some of the plots that we've seen, but we'll have more detailed information in the future on that. I would like to mention a really nice study that was just published here in 2020 by Rachel Brooks and others, where it was a similar study. It covered a larger area than ours, but they injected ailanthus trees at 13 sites in Pennsylvania and Virginia. They used the Virginia strain that had been found in Virginia and the Pennsylvania strain because it can't be moved across. Those can't be moved across state lines at this point. And so it covered 4 plant hardiness zones. There were more than 3,000 ailanthus trees. About 20% of those were injected with the verticillium wilt. And what they really saw was there was high mortality at all of those sites. It was progressing rapidly. They didn't really have any sites like our wild site where we had sort of lower levels of mortality. And they wanted to see if there was any limits by climate or stand factors that seemed to strongly affect the mortality rate of ailanthus. And those were really unimportant. Ailanthus, at all those sites, they were dying quickly, and those climate and stand factors were of minor importance to how the disease was progressing. And then I would like just a quick update. I don't know if there's a whole lot to report here on the EPA registration of verticillium non-alfalfa as a biopesticide, registering it with EPA. There is an effort that's being led by Dr. Raghavan Karudatan, who goes by Charu. He's with a company called Bioproducts Inc. In 2018, him and his team met with EPA and received basically a green light to go ahead and proceed with testing and putting together a package where this verticillium could be used as registered as a biopesticide. One of the good things, there's been a lot of research done primarily in Pennsylvania on non-target effects. Joanne has also done some of that in Ohio, which I didn't really touch on, but that can be used to bolster the case for registration. And they're going to do a lot of additional testing in Florida at the Horticultural Research Laboratory, Charu and his technicians. Not really sure of the timeline. I think it could be several years still before it's being able to be registered. It's at least to my knowledge, it's partly dependent on funding to support the additional testing that needs to be done on crops and other tree species as well. And with that, hopefully there's some time for questions. I'd be happy to answer them.
All right, Todd, thank you. So far, the only question we've gotten is how soon before we can buy it at Rural King? I think you answered that one towards the end there.
Yeah, no, yeah, hopefully sooner than later, but probably not within the next 5 years, I would say.
All right. Do you think the wilds has experienced lower mortality because it is more open landscape and thus a less ideal condition for the fungus?
That's a great point. We're not really sure yet. Those stands where the ailanthus was injected, I mean, they are a little bit more old field. We're on old strip mine land, but they are pretty dense forests. They're pretty densely forested. So that it could certainly be a possibility, and it might just be that the wilt that was injected at that time might have just lost a little bit of its. It might have not been as pathogenic for whatever reason. We're just really not sure. But that's a good guess.
And then we have a question. Did you indicate prickly ash is affected?
Oh, I did not. Thank you. Yeah, and there. We're also. I should mention a little bit. Joanne didn't touch on this much, but she's done some testing, and we haven't been as good about following through on some of her work on non-target organisms. But she also was looking at ginseng because it is related to devil's walking stick in the same family. And the preliminary results for ginseng were that it was not affected by the wilt, but I think there's probably still more testing that needs to be done.
How long does the fungus live after growing it in the lab? Like, how long do you have to apply it?
Yeah, it's a pretty short shelf life, and I'd have to go back and check on that, but it's more in the terms of several weeks rather than months. So it needs to be updated. But it can be one of the things that's been looked into is using to make more fungus is just going and collecting soil from infected stands, and then re-isolating the fungus from that soil has proved to be an effective way to get new inoculant. But it's a pretty short shelf life. I know that will probably be part of as it's becoming commercial in the coming years. That'll probably be part of it, is to try to find ways to increase that shelf life.
Okay, so if it is a native fungus, do we know what the host plant is here?
That's a good question. I mean, it's native. It seems to be cosmopolitan. No, I mean it's definitely found it's been found in other countries, and I think it's been spread around through agricultural crops to some degree. But really, the native here in the eastern United States, Ailanthus is by far the most susceptible plant. The devil's walking stick, prickly ash, there may be a few native things that could, are a host to it, but Ailanthus definitely has shown the greatest susceptibility.
Okay, and then we have an FYI, U.S. Forest Service, state and private, just funded Churu to continue host testing to meet EPA's request for more testing. So fingers crossed. And that came from
Okay,
Rick
great.
And we have a question of, can the spotted lanternfly transport the fungus?
Oh, that's a good. There might be somebody else out there that knows. I do remember some conversation about that. I don't think it's been determined yet, but there could be some more recent work. I don't have an answer for that, but I think someone has at least inquired about that, but I'm not sure.
All right, I think that's all of our questions. So at this point, thank you, Todd. We are going to try to get a speaker on the phone because it's not working. So go ahead and stop sharing there, Todd.
Okay.
Phil, are you able to join us by voice at least right now?
Are you able to hear me, Stephanie?
Yes, I can hear you.
Okay, then I'm guessing the rest of the audience can hear me too.
I am going to try and switch our screen here. Give me just a minute. All right, can one of my speakers please confirm that they see the ALB PowerPoint?
Yeah,
Yes.
You're not in presenter mode. You're in the other mode.
Okay, working on that.
We can see it.
Okay, so I think what we're going to do is, Phil, just let me know when you need to advance the slides once it starts responding again here. I tried to send this to Dave to share, but it was too big. So we're going to hope that this works. All right, I think we are good. So if you want to go ahead and start, Phil, just tell me when you need to advance slides, and I will advance the slides.
It's not showing up yet, Stephanie.
Okay, it just popped up on mine.
Okay.
So I'll wait, Stephanie, and give my introduction.
It's working.
Yeah, go ahead.
Okay. And just so you know, I do not see it in presentation mode yet. I just see the first slide and all the other slides on the left. But good afternoon, everybody. My name is Philip Baldoff. I am the program director for the Ohio Asian Longhorn Beetle Cooperative Eradication Program. I've been here for 8.5 years. Joined the program shortly after the initial detection of Asian Longhorn Beetle in Ohio, which happened in June of 2011. If you see on this first slide, it's a cooperative program, and there are a number of agencies involved. This is not all of them, but these are the main agencies involved. With the USDA, Animal Plant Health Inspection Service, Plant Protection and Quarantine being a lead agency, along with the Ohio Department of Agriculture, and the Ohio Department of Natural Resources is also involved with the program. Please advance, Stephanie.
And Phil, if you are seeing from the audience side, you're probably going to see these on the delay that they see.
Yep, so I'll just wait till it advances.
Okay.
So the Asian Longhorn Beetle, also known as ALB or Anoplophora glabripennis, it's an invasive hardwood tree killing beetle. This slide shows the life cycle. Let me start with the adult, which is the black and white adult beetle on the bottom left side of the screen. Male and female beetles mate during the summer months. The female beetle will chew a oviposition site or an egg site into the bark of a host tree, insert an egg, 1 egg per site. A larva will hatch out of that egg and then go through several molts as it grows and continues to eat its way, creating galleries through the tree. In the late spring, ALB pupates and becomes a new adult and chews its way out of the tree to perpetuate the life cycle. Please advance, Stephanie. The exit holes that are shown on the left, or the emergence holes, are about the size of a dime. So it's a big beetle once it emerges. Part of the problem with ALB is that it has a wide host range. Unlike emerald ash borer, which has a narrow host range, Asian longhorn beetle can infest 12 different genera of host trees. That's what's known. Those are the known host trees at this time. This slide lists the host trees in prevalence of infestation here in Ohio. Those shown in yellow, we have not detected any infested trees of those shown in yellow. Please advance, Stephanie. And the next slide will show, we'll take this data, we'll take these host trees and include some data to go along with it that will kind of help you understand the numbers that support this this ranking. Click one more time, Stephanie.
I did. I'm just waiting for it to come up.
Okay. Yep, that's fine. There's some animation there. Okay. So the take-home, excuse me, the take-home message on this slide, there's lots of numbers presented up here, but the take-home message on this slide is that by far and away, the vast majority of ALB infested trees in Ohio, as well as in the other ALB programs in the United States at least, are maples. Now, to kind of work you through what these numbers mean, I'll help you understand what I mean by infestation level. An A-level infested tree only has egg sites and no exit holes, so no adult beetles have climbed out of A-level trees. A B-level tree has greater than 0 and 10 or less than 10 exit holes. A B-level tree is greater than 10, less than 100, and a D-level tree has greater than 100 exit holes. So not only is our maples the most likely trees to be found infested here in Ohio, if you look at the different infestation levels, we have only found D-level trees that are red maples, or sorry, that are maples as well. None of the other species that we've found infested here have been found in D-level trees, and the research supports that. Thank you, Stephanie. And the risks, keep clicking through these, Stephanie. There's several different slides here,
Okay.
Or several different pictures, but there's lots of risks due to Asian longhorn beetle. With the first being tourism. Lots of people love their maple trees in the fall, and there are fall peepers and kind of an industry out there of tourism that draws in money to areas for people to look at those trees. The sugar maple industry can certainly be impacted as well as the timber industry. Nursery stock. There's lots of maple nurseries out there. And trees obviously do a lot to improve water quality, air quality, and the quality of life. Click again, Stephanie. And just from my perspective, I will tell you, as we inspect properties and find trees that are infested, knock on the door and tell the property owner, hey, we're here to eradicate the Asian longhorn beetle, and sorry to say you got infested trees on your property. Sometimes we're met with skepticism and a challenge from the property owner who says, I moved here specifically for these trees, and now you're telling me I got to remove them. And that kind of gets at that question of quality of life. People love trees, and unfortunately the most effective tool to eradicate this beetle is to remove trees. So how did Asian longhorn beetle get here? It really starts with climate change. The beetle is native to Asia, and back in the Gobi Desert has been expanding at an alarming rate, some 1,400 square kilometers per year. And the Chinese government back in the 70s developed a program to try to halt the spread of the expansion of the desert. And what they did, they planted a lot of trees. The Great Green Wall is what they call that. And most of the varieties of trees were hybrid poplars used, which are a susceptible host to Asian longhorn beetle. And so again, we got a susceptible host in the native range of the ALB. And Stephanie might want to click now, but I'm not done talking on this slide. And then, and that time in the 70s was right at start of a major expansion of exports from Asia to locations around the world, including the United States. And, you know, they use a low quality wood to create their pallets. And obviously the trees that are infested with ALB produce low quality wood. So they started using that poplar material to create their pallet wood, and you know, which coincides with the expansion of international shipment of goods, and which led to all the different introductions. Stephanie, you can click again. You know, obviously I don't know if ALB came to the United States on this particular ship that's being shown, but you know, as all those containers are full of pallet wood that hold your Nikes and Levi's and Apple iPhones. Once those goods get offloaded, that wood packaging material, advanced Stephanie, sometimes gets dropped and disposed of, and ALB can climb out and start a new infestation. Once ALB has started a new infestation, then it can spread locally by humans. You can see infested firewood in the back of that pickup truck. And it can also spread on its own. Please advance, Stephanie. The beetle can fly. And this map shows an overview of the different infested locations, known infested locations in the United States, with the first being found in New York in 1996 in Brooklyn, followed by a detection in 1998 in Illinois, then New Jersey, then Massachusetts, then Ohio, and most recently, just this year in South Carolina, there was a detection, a new detection. I could give a separate presentation on South Carolina. I was there to respond to that infestation for 2 months this summer, but I can't go into detail there because we're talking about Ohio. So this slide here kind of presents, if you want to back up just one, Stephanie, there's a...
I don't know if it's going to let me without delaying everything
okay.
several times.
Okay, that's fine. I'll just skip that one then. So
Sorry.
when our surveyors, nope, no problem. Our surveyors go out and look for lots of different signs, as well as the beetle itself. But most of the time we are more likely to find holes in trees. Click, Stephanie. As the beetle, we talked about those oviposition pits. The female mandible, as she's chewing those pits, she chews a hole in that tree, and you can see on the sides of those holes, what we call mandible scars. So if you imagine a pair of scissors, you walked up to a tree and opened them back and forth on the tree, it would cause these distinct grooves. The female mandible does the same, and so we look for those grooves. We'll also look for exit holes, those where the adult beetle climbed out. We'll also look for frass, which is the insect excrement. As it gets displaced, it gets pushed out of the holes and collects in branch unions and in the ground. Stephanie, this is a movie. If you can play the movie, it shows ALB emergence. There should be a play button if you mouse over the bottom of that slide.
I'm trying.
Yep, I understand. I guess we'll have to come back to that movie some other time. So Asian longhorn beetle, the only known, or you can try again.
It started after I said that.
Oh, okay. Yeah, so what this shows, one of our surveyors was walking by a tree and just happened to catch it right at a time when an adult was on its way out of the tree. And he stood there and snapped a picture every few minutes and saw the beetle exit the tree. Very soon after that, the beetle was captured and killed. Claremont County is the only known location in Ohio to be infested. There have been 3 different quarantines in Claremont County, 2 of which have been declared eradicated. Those are the ones shown in blue in Monroe Township and Batavia and Stone Lake townships. Tate Township and East Fork State Park and a portion of the East Fork Wildlife Area are still quarantined, as there are still infested trees there. This map shows where we have been surveying, with the different colors representing the ground where we have surveyed multiple times, with the green being just once, blue twice, 3, and orange 3 surveys. Click, Stephanie. Those other buffer lines are how far away you are from known infestations. All the orange dots here show where we have found infested trees, and we continue to find infested trees even now. And if you could click, Stephanie. And this slide kind of gives you an idea of the numbers that we're dealing with. So this shows the number of standing infested trees. So we have surveyors that go out and detect infested trees, and then we have crews that come follow those surveyors and remove those infested trees. And you can see it kind of goes up and down, up and down, up and down. And has never gone as far up as previous spikes have gone, and that's what you would expect as we survey in heavily infested areas. We don't get them all, and then when we come back, we'll find some more. So that's what we're doing. If you could back up one more slide, Stephanie, to show that last table.
Can we come back to it at the end? Sorry.
This is the end.
This is the end. Okay. Well, there will be a delay. Are you talking about the program numbers table?
Yes.
Okay.
Yeah.
Sorry, I was trying to gauge so that there wasn't quite the lag.
I would have done the same thing.
Well, while we're waiting, I did get sent a text question. In the mid-stage
Okay,
larva, is there a way to distinguish it from other wood borers in a piece of firewood?
No, not. You have to do some dissection under scopes or genetic testing. I haven't met anybody who's able to distinguish between ALB and other cerambicids.
Okay.
That's a great question, though. So this slide kind of gives another view of the program activities as far as numbers are concerned. Some notable numbers from this slide show the number of surveys we've conducted. We're over 3 million, which just astounds me. The number of infested trees detected were over 20,000. And we have removed over 19,400 of those detected infested trees. And that kind of helps you understand, again, what I was mentioning earlier, how we go and find some, and then we go and remove them, and then we go find some more, and we remove them. So we're kind of, our removal folks are constantly chasing our surveyors. We will also remove what we call high-risk host trees. So these are trees that we don't necessarily see signs of infestation in, but due to their close proximity to infested trees, they are a high enough risk of actually being infested that we will remove them or conduct chemical treatments on them, even without those signs of infestation. And we've removed over 86,000 infested high-risk host trees, and we've also done some chemical treatments as a prophylactic in those 2 satellite quarantine areas that I mentioned earlier in Monroe Township and on the border of Stone Lake and Batavia Townships. And then we also have had a couple cooperators assist heavily in a replanting program. ODNR has taken the lead on one of those replanting programs, as well as USDA Natural Resources and Conservation Service. They also assisted with replanting under their EQIP program, which goes a long way to help soften the blow when property owners lose a high number of their trees. Stephanie, if you want to click one more time, and then I'll be wrap it up unless there are other questions.
I think we had the credits up once, so if you don't mind, I'm not going to put it back up again because we are running behind schedule here today, and
Okay,
we may
fine.
come
Thank
back
you very
to
much.
questions at the end
Okay.
because I've lost
That
access
works.
to my Q&A box.
Okay. Thanks, everybody. Sorry
Thank
for
you,
the
Phil.
difficulties.
No, we got it to work, so we're okay. And I'm going to go ahead and have Dave Apsley share his screen. Oh no, are you guys able to hear me?
I
I
hear
can
you,
hear you.
Stephanie.
Yes, we can hear
Okay.
you.
My Teams just froze. So
I
I'm
see it
trying.
says your network is causing poor call quality is what we see.
Mine just went completely black.
Hmm.
Dave, are you able to share your screen?
I thought I did. Do you see it, Todd?
Yeah, I can see Dave's screen. Yep.
Okay, well, we're not
You're
going to have
seeing
Dave's
it in
video right now, but
presentation
just
mode.
go ahead, Dave. Hopefully mine responds.
Hello, everybody. It's Dave Apsley joining you from Jackson County. I'm a natural resource specialist with OSU Extension, and I'm sure I know most of you out there. I'm going to just give you a little update on sudden oak death and a response that we did down here in Southern Ohio in conjunction with a bunch of partners. So we'll move forward with the slides, and I don't think there'll be as much of a delay here, but we'll see. Except I got my mouse on the wrong page. So I love this quote I found from Georgia, and being a Georgia graduate, I thought I had to share it. Sudden oak death is actually a misnomer. It isn't sudden, and it doesn't just affect oak trees. That kind of sums things up. I wanted to thank Tom Macy mainly for sharing his slides, and then a report that Tom shared with me that Dave McCann from ODA, Division of Plant Health, shared that gives the timeline and the update of sudden oak death here in Ohio. And I probably shouldn't say sudden oak death, I should say Phytophthora ramorum. So what is sudden oak death? It's caused by a fungus-like organism. Technically, it's a water mold called Phytophthora ramorum. A lot of people are confusing it with the fungal disease oak wilt, and there have been no known confirmed cases of sudden oak death in oak forest in the east, as far as I know. So a lot of people interchange these, and I want to make it very clear that we are seeing oak wilt, but we have not seen sudden oak death. It was first discovered in 1995 in California. Really don't know the origins, but it is likely a non-native species. And basically, like other Phytophthora, it swims, not technically, but it actually like swims to the roots of the host plants through water. So you actually have to have saturated soil conditions for Phytophthora to move. And it's caused extensive mortality to oaks and tan oaks in Northern California and Southern Oregon along the coast. So here's a map It's a bit dated, 6-8 years old now, but that's a map of infected areas with Phytophthora ramorum. What makes this plant or this disease a problem is it affects many genera of plants. Not only the quercus, the oaks, but tan oak, lilacs, camellias, rhododendrons, vacciniums, viburnums, and so on and so forth. And it doesn't kill all the plants. So there's a pretty big risk and a likelihood it can be moved in nursery stock. And there have been multiple introductions in the East over the years. Probably one of the most significant ones was the early to mid 2000s in Georgia. But there's no evidence of it established in forest in the eastern United States. In Georgia, they have detected it by some of the stream dating we're going to talk to you a little bit about later in the streams, but they have not been able to locate it or isolate it in a native forest. It has become established in nurseries down there, and since I think they've been eradicated. As someone who works in Southeast Ohio, when we started hearing about sudden oak death potentially being brought into the state, we were a bit concerned because we spend a lot of time doing oak management work with an interagency forestry team in Southeast Ohio. And this is a potential risk map for sudden oak death. And you'll notice the high risk areas tend to follow the Appalachian mountain chain. And I really believe that those high risk are largely driven by the other host plants that are out there, things like rhododendrons and mountain laurels in the understory. But you'll notice when you get into Southeast Ohio, and I apologize, the border of Ohio doesn't show up, but it's somewhere along here, you'll see some of those moderate risk areas bumping into Southeast Ohio. So many of you may or may not remember, but back in July, Ohio Department of Agriculture put out a press release, and it came through our extension channels and through forestry channels. Looks something like this, and I'll share some of the details in the next slides coming up. So don't worry about all the details on this slide, trying to catch them all. In March, there was a shipment of plants from Park Hill Nursery in Oklahoma. I think the plants originated in Washington to Walmart, Rural King stores in at least 18 states. Phytophthora ramorum infected plants were first noticed and reported in Indiana in May. Several rhododendron and lilac varieties were confirmed infected. In Ohio, we were part of those original shipments. They think plants were sent to about 300 stores. And of the varieties that were, quote, hot, that potentially had disease or were tested to have the disease, there were 156 stores that received the plants and about 1,600 plants. By June, all the plants were pulled from the stores that were still there, but in many cases, plants had already been destroyed or sold. So some of the plants did get out and were sold to the public. So with the press release, there was a request for people to report if they had, let me skip back here. From June to October, the plants that were collected were tested, and they could just test for Phytophthora in the early test. It's called an ELISA test. And of those 60 positive tests that showed they had a Phytophthora, 37 were confirmed to have Phytophthora ramorum. July and August public service announcements went out. A web page went up on the ODA site and started asking folks to report and send in their plants. So in that time period, about 21 inquiries were submitted. And to a little effort we're going to talk about here in Southeast Ohio, there weren't any or many of those sites down here other than the original store locations where these plants were found or reported. So in August, a group of us working in Southeast Ohio decided that we wanted to try to help out and pitch in a bit. So we actually worked with ODA and we identified, trained, and outfitted some local points of contact. So we found folks from Soil and Waters Extension and Division of Forestry in the 17-county priority oak management area in Southeast Ohio. And so we did an online Zoom training, one little practice for this COVID session, I guess, to do training by Zoom. We had, I think, 38 people attend, and then we provided them with materials that they would need to go out and actually help the landowners collect and destroy the plants. We also created some local press release and put the local points of contact. We felt like down here in Appalachian, Ohio, folks were more likely to call their local soil and water extension office than they might be to report to a website in Columbus. And we had our local folks respond to 6 of these inquiries. So we actually had people call in. There were 6 inquiries, and all but 1 tested negative for Phytophthora ramorum. The one site that tested positive was collected by a soil and water staff member, and ODA sent out their staff to go ahead and collect plants from that site, and then they also removed the plant and drenched the site with fungicide. They also collected additional plants from the site, and those plants tested negative. So it was an effort to get the word out to Southeast Ohio, where we've got a predominance of oak-dominated forest, and we had a bit of a response. Another little thing I wanted to point out is there was another potential introduction a bit later. Some roses came into Walmart locations in October, and 12 of the 13 samples that were collected came back negative. There was one inconclusive test up in Lorain County. And then to wrap things up, since we are certain Phytophthora infected plants were brought into Ohio, there's an effort to actually do some monitoring to see if we can pick up Phytophthora in the ecosystem. So Division of Forestry got a grant, and they're doing 2 rounds of sampling at 7 to 8 sites in eastern and southern Ohio. They're sampling sites downstream of potential introduction sites, predominantly Rural King and Walmart stores on perennial streams. The sampling has to take place when the water temperatures are between 59 and 77 degrees, so there's another sampling period coming up as things cool down. So what's happening is water is collected near the stream bank, and then host material from rhododendron leaves is placed in the bottles and incubated according to protocol. Samples are then sent to Penn State Pathology Lab for analysis, and the first round of sampling has taken place, and so far there are no results. So again, Tom Macy is leading that effort with the Division of Forestry. Tom, you're welcome to, if we have time for any questions, to join in on that part of the discussion or add to that if you'd like. And with that, that's all I've got. So Stephanie, it's up to you as to whether we have time for questions.
I think we probably better hold off on the questions until the end, and then if people have them, we can stick around a little bit later. Sorry, we are running a little bit behind here. We do have one more we want to get to real quick about how to get pesticide credits. So I know that's usually a big highlight of the Forest Health meeting. So we have Mimi Rose available to help explain how to get your pesticide credits. Mimi, are you able to share your screen?
Yes, I'm working on that right now.
Okay.
Okay, are you seeing it?
You should be good.
Okay. All right, yes, I'm going to give you a very brief update on how recertification is happening in this year of COVID-19. Okay, so while ODA is having to make accommodations for pesticide recertification, obviously there's been very few, if any, live programs since March. They were able to resume pesticide exam testing. At this point, it is only available at their main location in Reynoldsburg, Ohio. So what about recertification? Well, I guess I should have introduced myself at the beginning here. I'm Mimi Rose with the Pesticide Safety Education Program, which is part of OSU Extension. And since the spring, we've been working with the Ohio Department of Agriculture to put together an online recertification platform. And actually, this has been available since well, August 10th for the commercial applicators and a little bit earlier than that for the private pesticide applicators. So initially, this has been made available only for those who weren't unable to recertify for the 2020 cycle, although it looks like this is going to be extended for 2021. By the way, if you are expiring sometime during 2020, just be aware that those recertification deadlines have been extended to December 1st, 2020. Okay, so what does this online recertification look like? We're actually using a course platform called Scarlet Canvas. It's a sister platform to the Carmen Canvas that they use for the OSU online classes. And what you're looking at there on the screen is what the main web page looks like. And it's organized, well, there's some initial stuff you really need to read so you understand how to complete the course. And then there's a set of videos for core, for category 1, 2, et cetera, et cetera. And what they are is videos that you will watch for recert credit. Where do you find this recertification online course? It's on the PestEd website, pested.osu.edu. And this page is where it looks like, or what it looks like. It's the jumping off point for the recertification platforms that we're offering. So there's one for private applicators, one for commercial applicators, and there's also for agricultural fertilizer applicators as well. Okay, so how do you do it? Well, step one is at this page to choose whether you need private or commercial pesticide recertification. Just FYI, private recertification is for those involved in agricultural production. Some of you may have private licenses. For example, if you're a woodlot owner, you could conceivably have an agricultural production, or excuse me, a private license. Probably most of you in this audience, I suspect, have a commercial license, especially if you work for state government and apply pesticides, you have to have a commercial pesticide license. Well, at any rate, step 2, after choosing the appropriate course, you actually have to enroll and register for the course. This indicates what the course fees. It's broken down by how many hours you need. You register, you make the payment, they send you a link, and then you have to go to that link to enroll in the course. So when you go through this process, you actually create an account and a password on the Scarlet Canvas platform, which allows you access to the course, and it allows you to return again and again and complete the videos at your own pace. So really the only constraint on you is when your recertification deadline is. You need to complete it by then. Step 4 is to actually go through the videos and complete your recertification. And I really have to emphasize that you're going to want to read the instructions carefully. We've done our best to make this as simple of a process as possible, but I have to admit it's still a little confusing. So there's even tutorials, video tutorials that show you how to successfully complete the course. So just a few tips. You only need to view the videos for the categories that you need to recertify in. So you don't have to watch every video. There are 26 commercial categories after all, so that's a lot of videos. However, within any given category or for core, you have to complete all the videos within that category. After you complete a video, you essentially sign online a statement attesting that you watched those videos. That's your kind of word of honor. FYI, we actually can and do occasionally go in and can see how much time the people spent on the videos. But at any rate, it is to some degree, it's an honor system that you attest that you watched the video. And then at the very end, you must complete your recertification form, enter your license number, and this is the data that will go directly to the Ohio Department of Agriculture. That form and that data in it goes to them. Okay, let's see what I'm just showing you here a little bit more of the main page and how you scroll down through it. and to see all 26 commercial pesticide categories. And that you can complete it at your own pace in multiple sessions. You just have to complete it by the deadline. Okay, just a reminder, once again, to make sure you choose the correct course depending upon whether you have a private or commercial pesticide license. Okay, well, that kind of sums it up for the online recertification platform. Any questions, you can also call our program. That telephone number is provided on our website and in the information on the course. So what about next year, 2021? Well, actually, we just talked with the ODA yesterday, and we do know that we will be able to offer the online recertification next year as well. What also has been discussed is the possibility of live programs, kind of like today's forestry program, and whether those may be possible to recertify online during live programs. So ODA is still really mulling this over. We actually completed kind of a small trial certification program for them this month and demonstrated how we could provide the data they need for that and also ensure to some degree that the people are actually watching during that time. We're expecting approval for some of the OSU extension programs that we do for research. They're also going to consider whether other Ohio organizations may offer research as well. So what the main kind of considerations for the ODA for their side of things is they want accurate, authenticated recertification data. So obviously when it's online, the people aren't going to be handing in those research forms. How can they get that information and have some attestation statement that the people actually were recertified? So we're actually working with ODA how we can accomplish that. And I kind of think if this is going to go to other Ohio organizations, they're going to need to follow the pattern that we are currently working on. So that pretty much is my update. If you have any questions, give us a call or check out our website. And once again, that online recertification program is available and up and running on our website right now. Thanks for your attention, and I'll be happy to answer any questions.
Thank you, Mimi. So far,
Sure.
we've only had one, and that is
Okay.
what if your certification has already lapsed? Can you do these recertification courses, or is there another online option for certification?
Unfortunately, if recertification has lapsed, I believe your only option is to retest.
All right, and that's the only one I see, so thank you.
Uh,
If any others
but
come
just-
up, I'll let you know.
Oh, Stephanie, can I interject one
Yep.
thing? Okay, just make sure you're not thinking that it elapsed on September 30th, 2020. Okay, recall that the deadlines have been extended. So you have, if yours was expiring in 2020, you have until December 1st. So I just want to make sure I got that point out there.
All right, thank you.
Sure.
Okay, so that should conclude our forest health meeting for this year. Again, I apologize that we've run a little late. I apologize that we had to do this online and for the few issues that we had, but overall it went better than I thought it might. So I'm very grateful for that. Just a reminder, if you need ISA or SAF credits, please make sure that you have sent me a message in the Q&A box with your name, whether it's ISA or SAF, and your member ID number so that we can get those credits awarded to you. Otherwise, if you come up with any questions on any of the talks, feel free to send them to me. I can forward them to our speakers and get them answered. I don't see any others that have popped up in here. Do any of our speakers have any last minute statements?
I don't, Stephanie.
Thanks,
All right.
Stephanie, for hosting. I know it was challenging, so you did a great job.
I can honestly say I'm very relieved that it's over and that we made it to the end and got through all of the talks. And thank you all for joining today. And I look forward to hopefully seeing all of you in person next year for the Forest Health meeting. Thank you very much.